Today we're going to have about 3 sessions, and the first obviously will be about the reference, HCA as a reference to disease. There are opportunities for us to discuss and collaborate as well, and to discuss the future directions of how HCA can help Africa in terms of futures of medicine. And then there will be closing panel discussions that we're going to have at the end of the day. In terms of housekeeping, again, from one session to the other, you need to sign out and then close, and then you get in by clicking on broadcast on your sign-in page on the agenda, so you can get into the next session. Please do not forget to join the social world, where we can have conversations and discuss with each other about the various presentations, as well as discuss aspects of future work that we could actually engage in to collaborate on. If you have your profile picture, please do not forget to edit your profile and add your picture, so we can put a name to the face or put the face to the name when we are actually having those discussions. So this definitely will be good if everyone can do that. There are resource pages, and this is not just for students, it's also for everyone else, including some of us group leaders and PIs. And there you can find a white paper and several publications and videos, and of course the HCA's commitment to humanity. There is a help desk. If you have any issues, please contact the help desk or check the fact sheets, where you can get more information and get some help and orientation on what you should do. When the presentations are over, there's some time for chatting and for questions and answers. If you want to put up your questions, you can put it in the chat, but there's also a question and answer icon. You can use the question and answer icon where you can type in your questions. If you don't want your name to be identified, you can type in as anonymous before your questions. You can also put your questions in the chat box, and this will be read out. You can raise your hand as well during the period of the QA, and you're given the opportunity to answer, to present your question. To make interactions easy, the app for the Bizabo is available. You can download it from Google Store, or you can also download it from Google Play or App Store. So I again thank the organizers. I didn't introduce myself properly. My name is Alfred Amambua-Ngwa. I'm actually a resident at MRC in the Gambia. I am a group leader. I'm mostly working on malaria, but I also have several projects, some of which cuts across human genetics. We are interested in several aspects of disease beyond malaria, and how infections also interact with non-communicable diseases such as cancers and other systemic morbidities that are inflicting our populations. We're hoping that with genomics tools and several other new emerging technologies, including single-cell sequencing, for example, in the case of HCA, we should be able to further understand the diseases and also be able to help with the development of new interventions against these diseases.
So we know TB continues to be a public health concern. There's a healthy TB vaccine pipeline, and we urgently need a TB vaccine. As shown here, there's different vaccines that are currently in development in different phases of clinical trials. And those vaccines include adenovirus, adenovectored vaccines, as well as recombinant BCG strains and subunit TB vaccines. However, while that is very promising, and I think the TB field started to feel a little bit maybe less motivated prior to this ARIS-C 0404 prevention of MTB infection trial, which results were published in 2018. And this really was a rejuvenation and a re-energization of the TB field. In this trial, adolescents between the ages of 12 and 7 were randomized to 3 groups. One group received a single vaccination of BCG. All these participants had received BCG at birth, so this is a BCG revaccination trial. The second arm of participants received H4IC31, a subunit vaccine at 2 time points 56 days apart, and there was a placebo group. All adolescents at enrollment were quantiferon negative, suggesting that they were not exposed to MTB. Participants were followed up for a period of 3 years with the quantiferon, a proxy of MTB infection performed every 6 months. When the results came out, as shown here on the right, we looked at the number of participants who had become infected and found that there was no vaccine efficacy. So there's no difference in those who had received vaccine and who hadn't in terms of the number of people who became infected in this trial. However, the secondary endpoint for this trial was what is known as quantiferon conversion or sustained quantiferon conversion. These are participants who test positive and then remain quantiferon positive. What we noticed in this trial was that those who had received BCG had a reduced rate of sustained quantiferon conversion, and this vaccine efficacy was shown to be 55.4%, which was really promising. While this trial was happening, the HVTN designed a smaller experimental study known as HVTN 602. It had a similar study design as that shown previously, the ARIS 040. In this trial, it was much smaller. As you can see, only 24 participants were assigned to the BCG group and 24 participants to the H4 group. For today, I will not share any data for the H56 group. I will focus on showing you the data we generated by doing the intracellular cytokine staining assay shown here in red, where we had PBMCs collected from day 0, day 14, day 70, and day 168. And for those in the BCG group, we collected blood at day 28. We also performed single-cell proteogenomic profiling at day 0 and day 70. This is just to show the 28-color ICS panel that was used. So we were fortunate enough to get a FACS symphony, which would allow us for the first time to really look at 28 parameters on a single cell. So we designed a panel that would allow us to look at not only at T cells, which is very commonly done, but to also look at non-conventional T cells such as gamma deltas, mates, NK T cells, and to look at cytotoxicity. So the first thing we looked at is, well, what do these vaccines do immediately after vaccination? So shown here are the CD4 T cell responses after vaccination. A response was defined as the production of any one of the 9 cytokines highlighted here, being gamma, IL-2, TNF, IL-17, IL-4 or 13, IL-22, CD154, granzyme A or perforin. Shown in the dark red are responses to TB whole cell lysate. This is a lysate from MTB H37RV, so it really tells you responses not only to BCG but to all TB strains. We also then show the responses to BCG Pasteur shown in the pink, as well as responses in the H4 arm to the vaccine-matched PEP proteins, antigen 5B and TB10.4. What you can see after BCG revaccination shown in red and in the pink, there was a significant increase in the responses in the BCG arm after revaccination. But you will also note that these responses were present at baseline. At day 168, we found that these responses remained elevated above pre-vaccination. In the H4 arm, only you see an increased response to antigen 85B 14 days after the first vaccination, but only two vaccinations are required to induce a TB10.4 response, as seen here at day 70, and these responses remain above pre-vaccination, really suggesting the requirement for 2 doses for this vaccine. We were also interested in knowing, well, what memory responses are induced by this. It's believed that a long-term memory is important for sustained efficacy. So we looked at the responses in the antigen-specific cells. What we noticed, was that at baseline, already in those who had BCG, there were naive-like cells, central memory, effector memory, and terminally differentiated cells at baseline, suggesting that pre-sensitization was playing a role here. We also noted that there was an increase in naive-like central memory, effector memory, and terminally differentiated cells in the H4 vaccination, and in particular to the antigen 85B peptide pool. We next wanted to use unique ways of really trying to get into this data and really use all the markers that we had to look at these data. So we used UMAP, where we identified 18 unique clusters of CD4 T cells, as shown here. Shown in the right, you will see the combination of markers expressed by the different subsets. I just want to point out, while this is a lot of information, that we found that the dominant responses were CD154 and TNF-alpha co-expression. We noted very little expression of IL-22 and a complete absence of HLA-DR, which many studies have shown to be important in terms of progression to TB disease. We then wanted to say, well, generally, what was the trend in terms of change in responses? You'll note in the BCG arm, shown on the left here, that we saw no changes in the clusters that we had identified to antigen 85B and TB10.4, except for cluster A. Overall, many clusters changed in response to BCG or TB whole cell lysate in the BCG arm, and as expected, in the H4 arm, responses were generally only changed after vaccination in the H4 arm in response to antigen 85B and TB10.4. When we wanted to understand what these clusters really looked like, we looked for clusters that were changed after vaccination. What we found were 5 clusters shown here that changed over time. Cluster A, which has been identified and reported on before, was a Th1 star, which is a CXCR3 positive, CCR6 positive cluster that expressed IL-2 interferon gamma, TNF, and CD154. This cluster was again identified as cluster B, which was Th1, but in the absence of IL-2 production. And the predominant responses were effector memory responses, both in the BCG and in the H4 arms. So we also had done site-seq or single-cell proteogenomic profiling to better understand these responses. We performed profiling on PBMCs from day 0 and day 70 after PBMCs were stimulated for 16 hours. We had 17 participants in the BCG arm and 8 in the H4 arm. This work is done in collaboration with our sister lab in Seattle by Lamar and our computational biologist, Val, who's really led the efforts on the analysis, as well as Andrew Fury-Gartland and Erica Anderson and Evan Newell. In order to look at antigen-specific cells, we did sorting where we sorted 69 positive, 154 positive CD3, CD4 T cells, as well as CD69, CD137 positive, CD8 T cells. We also collected the supernatants from these stimulated to look at secreted factors. We had a panel of 109 antibody-derived tags used to assess protein expression. Overall, we had obtained a high-quality data post-QC of approximately 320,000 T cells. So we first did an analysis just looking at pseudo-bulk changes in these cells. So as previously described, the CD4s were CD69 and 154 positive, and the CD8 T cells were CD69 and CD137 positive. What we found was that there was significant upregulation of TNF family receptor S4F, otherwise known as OX40, and there was a trend to see an additional or increase in the expression of TNF, interferon, and granzyme B, confirming what we were seeing with the ICS. But this finding of the OX40 really suggested that this is potentially a protein that we should look for in further studies. We also then looked at the individual site-seq, and what we did for this, we use the weighted nearest neighbor workflow, and what it does, it looks at it independently processes and does dimensional reduction based on each modality, since we had protein expression with our ADT as well as gene expression. It then takes the 2 modalities and integrates them and does a downstream analysis as shown here. Using our site-seq, we were able to identify 19 clusters of T cells, looking at protein and gene expression, and mainly 2 big clusters came out, the CD4s and the CD8s, as this is how we sorted. When we delved deeper into these clusters, we identified both CD4 and CD8 T cells, but also some mate cells, gamma delta T cells, and NK T cells shown here in 17, 18, and 19. We then wanted to understand, well, where do our antigen-specific cells fall into? And look, shown here on the right, you can see where it is a lighter green or blue is more cells. And so we noted that most of our cells, as we had expected, fell into, as we had enriched for them, into the CD4 T cells, into a naive-like cluster of T cells, as well as into mate NKTs and CD8 T cells. We then asked, well, what does vaccination do to these cells? We shown here is the BCG arm in the top and the H4 arm in the bottom. These were the 4 clusters that changed over time, and we noticed a decrease in naive-like T cells, which was accompanied by an increase in effector memory CD4 T cells in the BCG arm. We also identified significant changes in the mucosal-associated T cells as well as the NK T cells in the BCG arm. Similarly, in the H4 arm, we noted a decrease in naive-like CD4 T cells associated with an increase in effector memory CD4 T cells. And not surprisingly, we found that there were no changes in mate and NK T cells in this arm. What was particularly disappointing, but still very interesting, was when we looked overall how many genes were differentially expressed in these antigen-specific CD4 T cells. Shown here are the BCG arm and the H4 arm. Very few genes were differentially expressed in the antigen-specific CD4 T cells after BCG vaccination. And in fact, we only saw 1 change in the mucosal-associated T cells with an increase in leukotriene B. When we looked in the H4 arm, we saw some more changes, but still very few, with 4 genes associated with an increase, 3 increase, and 1 decrease in the H4 arm. 5 genes that were changed in the effector memory's CD4 T cells and 7 in the NK T cells. I think we were really surprised to see how very little had changed when we looked at these data. So in conclusion, I think efforts are ongoing to define responses to TB vaccines. And in particular, in this study, we were hoping that we could identify correlates of protection from the smaller study for the ARASEO-404 trial to explain the efficacy that was observed. The BCG revaccination showed that it boosted pre-existing responses, even though these adolescents were quantiferon negative. And then the use, which I didn't show, but the use of BCG and MTB lysate actually captured very different responses, and it's something to think about when doing TB research and designing of experiments. All this work, though, is part of a bigger set of work, which is the TB immune correlates working group. And I won't dwell on this, but you can see there really are a suite of assays that are being done with collaborators to really try and identify what the correlates are in the ARAS-040 study. And so our data will feed into that scheme. So finally, I just want to acknowledge the people who are involved in these studies. So this is our group here at the Cape Town HVTN lab, led by Erica Anderson-Nissen. And just to say thank you to all the protocol team and the sponsors, as well as the community advisory boards and participants of this group. Thank you.
So this is going to be a complete change of gear from the previous talk. I'm going to speak more about principles and policies associated with the Human Cell Atlas. This has to do with equity and our commitments to humanity. This entire talk has been coalesced by the co-chairs of the equity working group, but also has been value-added and endorsed by the organizing committee and the ethics working group of the Human Cell Atlas. The 3 co-chairs of our equity committee are Musa Malanga, Alex Shellick, and myself. And on behalf of all of us, I'm making this presentation. I'm going to start with a warning that was given several years ago, 12 years ago, by Charles Rotini and Lynn Jordy, where they said that genomics provides novel insights into the causes of and susceptibility to disease and adverse reactions to drugs. And they warned that genomics is a double-edged sword. If used carelessly, genomics may exacerbate disparities. And this is where we are concerned, and this is where we are coming up with principles and policies. And so essentially what we want to do is to use genomics in a more precise way. And our belief is that precision genomics can actually promote health equity and not exacerbate disparities if we are careful enough from the beginning while we are embarking on these studies. I'll just give one example where precision genomics has actually helped, and we are talking about CML, which is a cancer of a special kind of cancer of the blood and bone marrow. And it usually does not afflict children, but when it does afflict children, it's devastating. In the 1960s, about four children survived CML, but because of certain understanding of the disease based in genomics today, in 2017, 92 of the 100 children who get afflicted with this kind of cancer survives, and today it's even more than that. The understanding came from various kinds of molecular studies and genetic studies, identified the genetic alteration that leads to a fusion protein. It's a translocation from a small region of chromosome number 19 to chromosome number 22 that leads to production of a fusion protein called BCR-able fusion protein. This particular protein has negative impact. However, if it is inhibited, and because of this understanding of genomics, tyrosine kinase inhibitors target this particular protein, and its impact can be completely obliterated. So this was the understanding from genomics that came, and today lots of the children actually survive. Unfortunately, there is also a bad news associated with this, that some patients become unresponsive to tyrosine kinase inhibitor therapy. And the reason was unknown for a very long time until more recently, about 5 years ago, when at least one set of understanding or one kind of understanding came about, again through genomic studies. And what it turns out by studies of multiple children with this kind of cancer, a set of somatic mutations that associate with response to tyrosine kinase inhibitors. And these are somatic mutations, these are not germline mutations, and therefore it arises in these individuals, in these children, and are not passed on, but they afflict these children in adverse ways that therapy does not actually work. So increase in genomic precision by understanding these kinds of somatic mutations actually improves disease management. And these are children, I work on cancers, and these are children in the cancer hospital. They are performing on the foundation day of the cancer center, and they're quite happy, and these are all children with this kind of CML. This is the Saroj Gupta Cancer Center and Research Institute in the city of Calcutta, and that's the institute. And we are very happy that this precision understanding can actually make these children so happy, and they are able to work on their daily activities and enjoy their lives. One of the clearest messages from genetic studies is that cancer and many other diseases are much more biologically heterogeneous than originally recognized. And this heterogeneity leads to heterogeneity of clinical outcomes, leads to difficulties in management, responses to therapy, and also impact on survival. So it's important for us to understand the heterogeneity at the molecular level, and the human cell axis is actually enhancing our ability to understand the kind of heterogeneity that we have in these diseases. And this actually leads from a uniform therapy across the board of patients to risk-stratified screening and treatment, reduction of overdiagnosis, more personalized intervention, and that in turn leads to saving of expenditure on therapy, overall increase in the number of years of disease-free survival. So this understanding at the single cell level, the understanding of heterogeneity indeed helps. So the detailed molecular understanding of diseases in relation to normal health is impactful. Human Cell Atlas project is essentially trying to understand normal health. I'll skip this. It's currently the pinnacle of precision genomics. We're trying to define the human body cell by cell, understanding human biology at the deepest molecular level. Eventually, the information will enable even better disease management, will save on cost of treatment. Just giving you an example of that, reduce patient suffering, reduce anxiety of family members, and increase patient survival. So these health benefits, if they are conducted well with a proper understanding, proper planning, the health benefits that will accrue from this understanding of human biology will accrue to across geography, ethnicities, gender, and ages. That's what we believe is equity, and this must be thought through very carefully and planned very carefully. If we look at this particular graph that comprises various kinds of diseases, asthma, coronary artery disease, childhood obesity, etc. On the x-axis, we have the number of people affected in the US, and on the y-axis, we have a disparity ratio measured in a particular way. And what we see is that there is a high amount of disparity even when the number of people afflicted with the disease is not so high. This calls for a necessity, or emphasizes the necessity, of promoting research considering social, environmental, and genetic factors. And we need to do that to ensure that all individuals receive the right diagnosis and the treatment at the right time. And what we believe is that if this is conducted in a socially just manner, such that the rising tide of precision medicine lifts all boats, including groups that are disproportionately affected by the disease. And if you look at asthma, for example, where there is a high disparity, there is evidence supporting an association between air pollution exposure and incident childhood asthma, and that's inconsistent. The association is inconsistent and may actually depend on genetic factors. It's not just the extent of air pollution that's positively correlated with incidence of childhood asthma, but it's also dependent on genetic factors. So these environmental interactions are important for asthma development, and there's already some evidence of significant interaction of certain genes with air pollution. Genomics actually is falling on diversity, and this is the title of a Nature commentary published about 5 years ago, which reviewed a large number of GWAS studies. And as a result of that, what we find is that it is impacting on various kinds of disease classes that one is dealing with, whether it's responses, whether it's autoimmune diseases, aging-related diseases, or even responses to various kinds of drugs, particularly anti-cancer drugs. There is considerable variation across populations, and actually ignoring these variations will result in health inequity. And so what in HCA, the idea is to be inclusive, and inclusivity is a cornerstone of HCA. The questions in cancer biology that are addressed by emerging directions with single-cell RNA sequencing are manifold, and we have learned various kinds of things through HCA's involvement in understanding normal human biology and also tumor biology, and this is providing us with greater insight into the formation of these tumors and how they develop, and as a result, they are actually helping us manage cancers better. The HCA roadmap is therefore to organize this in a collective way, in an inclusive way, study various systems, organs, tissues, regions, etc., and collect tissues from various sources across ethnicities, across genders, across age groups. These tissues are then both experimentally and experimentally data are generated, and computationally the inferences are drawn from the data, and they are coordinated and organized in a major way through collaboration of funders, working groups, meetings, and so on and so forth. To promote equity, HCA is actively taking actions to share knowledge of concepts and technologies by organizing workshops and roadshows in low and middle-income countries. It's empowering scientists from underserved and less endowed countries to participate by engaging them as partners in research and translation, providing access to technologies, educating and building capacity, and in turn, people who are the leadership in HCA is being educated. We are also enabling provision of less expensive and more portable technologies and reagents preferentially to the low and middle income countries. The commitment of Human Cell Atlas to Humanity is a set of principles that have been recently enunciated, and this was the initiative was taken by the Equity Working Group. It subsequently been endorsed by the Ethics Working Group and also by the Organizing Committee. What we are studying, like I said, that we are studying cellular variation in healthy individuals. There are many different cell types in the human body, even in the same organ. Proportions of cell types can vary between, for example, males and females, children and adults, smokers and non-smokers, Bantu speakers of Africa versus Austroasiatic speakers of India, etc. So it's important to recruit individuals across these diversities, and the way that we recruit individuals, we recruit them through self-identification, but not by doing any kind of helicopter science. The focus will be, or is, on local needs through local collaboration. HCA membership is open to scientists globally. We are embracing, consulting, and working under the leadership of local scientists in local laboratories to the extent feasible. The samples that are generated are actually being analyzed in the local laboratories. The data storage, analysis, and sharing of results are being done again locally. It's stored in publicly accessible global databases, including and ethical standards, including local rules and standards are being followed. Personal identifiers are, of course, stripped from these databases. Collected data are analyzed using statistical and other bioinformatic methods. Local HCA investigators or other persons in the community are encouraged and trained to participate equitably in data analysis. The data that are being used, we recognize as potential for discrimination, and we explicitly state that race is a social construct and not a biological one. Therefore, we strongly oppose the use of these data on biological differences. These data show that there is a lot of diversity, but we oppose the use of these data on biological differences for social discrimination. How do we ascertain whether we are making progress in enhancing equity or in promoting equity? We are developing a set of metrics to measure and track over time. We are developing metrics to measure diversity of investigators who are participating in HCA, diversity of representation of donors of tissues and samples to HCA, the extent of populating the various databases with anonymized data from diversity of groups, and local benefits that accrue from participation and data. We believe that these metrics will actually help us track over time whether we are improving in terms of equity or dissemination of equity. Precision genomics is poised to provide the right care to the right individual at the right time to advance health equity, and HCA is advancing precision genomics and health equity. We have actually the principle that I just enunciated, that is not yet available in the public domain. It will soon become available in the public domain. But we have learned certain lessons as we are discussing human cell equity and human cell atlas jointly, and the lessons learned has already been put together, and this has appeared a couple of years ago as a comment in Nature Medicine, and this is open access and is available. So I wish to also finally inform you that equity is being promoted in multiple parts of the world through engagement of local scientists by way of training and by way of holding workshops, roadshows, etc. Several of them have been held. There was an initial equity strategy meeting in England, followed by a roadshow in Brazil, followed by another in Ethiopia. Most recently, even during the pandemic, when we were thinking of holding a hands-on training workshop in India, it could not have been, was not held because of the pandemic. It was held online, and this was organized jointly by our institute and the MIT. There were over 200 participants, and there were people from multiple countries participated in this online workshop, and the countries included Bangladesh, France, Germany, India, Pakistan, UK, and the US. There is another roadshow that's coming up very soon, which will be in Vietnam, followed by another in Saudi Arabia, and funding agencies are actually supporting us in holding these workshops and promoting equity and local participation in the Human Cell Atlas project. Thank you very much for your attention.
So my name is Henrik Kloverpris. I'm a faculty member at the Africa Health Research Institute in South Africa. And I'm going to present to you some of our new data we collected and analyzed recently. So it's really the first time we are presenting this data, and we're looking at sort of the activation of innate lymphoid cells in HCA-infected lymph nodes. But before we get onto that data, I just want to show you basically, here's ARI here in Durban, where we process and have really access to some fantastic infrastructure that allows us to perform some live analysis on live tissue from nearby hospitals. So the implementation of single-cell RNA sequencing in my lab has really been due to a long-standing collaboration with Alex Shaleb and his team in Boston, and particularly input to this project has been from Emmanuel and Rabia, who's been setting up the SQL, the high-throughput single-cell RNA-seq platform in my lab in ARI, processed by Jens Akili and Elvira, and really with support from Sam Kaiser and Sarah Nyquist, who's been excellent collaborators in supporting us in this work. And Nicholas is really a new student who has been taking these data on and been doing a really good job in analyzing this data set. So our location in Durban, many of you would know, obviously, where the global AIDS epidemic is localized. And I think this map shows you clearly the importance of the work we're doing in ARI and in sub-Saharan Africa, particularly on the eastern part of South Africa here. And we can see, if you zoom into this region here, this is one of our population surveillance sites area where we have very high prevalence of not only HIV infection, but also the presence of viremia in 1 of 5 people. So in these red areas, if you just go in and sample one, any particular person, 1 out of 5 would have detectable viremia in the blood. So HIV infection is really not necessarily a disease of the blood, but really a disease of the tissue, and particularly the lymphoid tissue. So here's a proposed study or sort of outline from Steve Deeks a couple of years ago that highlights, the impact of the GI system, particularly the breakdown of the intestinal barrier in the large intestine, leads to these microbial products that then gets into the not only circulation, but also becomes activated of the other parts of the immune system, and particularly some of the myeloid populations that can that can sense these microbial products leads to this activation of monocytes, macrophages, and that can then lead to activation of other immune substances that are going to come back to it in just a minute. That really, all of this kind of leads to vascular dysfunction, tissue necrosis, fibrosis, and a mechanism that we call multimorbidity and immune aging. So therefore, we're really interested in studying what happens not only in the blood compartment, but really what happens in the lymphoid system, and particularly the lymph nodes that are draining some of these compartments here. So some of the cells that are really important in maintaining tissue homeostasis is the innate lymphoid cells that here is pictured alongside the CD4 T helper cells, and you can see that they really reflect the different CD4 TH1, 2, and 17 subsets that shows that they have similar transcription factors and also similar cytokine profiles in these ones. But really, not much is studied, particularly in HCA infection, on these subsets here, and also very little is known, particularly in these lymphoid tissues in human in general. So we showed a couple of years ago that actually HCA infection has a massive impact of these subsets in circulation, and we showed that successful treatment to undetectable level doesn't bring these ILC subsets back in circulation. Other people found similar results. Even fully suppressed individuals don't restore these ILC subsets in HCA infected individuals. So one hypothesis that actually Nicholas came up with here, that the general inflammation in HCA infected tissue could actually impact on the so-called transdifferentiation of different ILC subsets. And this is work that is been driven by Hagen Spitz group in Amsterdam, where they showed that you can manipulate these ILC subsets to become either ILC1s or ILC3s, depending on what kind of signal you give them. And the same is true for even for the NK cell subsets that can transdifferentiate into ILC1 subsets. And this can really change the inflammatory environment within these tissue resident cells. And here's another mechanism where we think that potentially production of IL-1 beta, IL-23 can impact on your ILC3 subsets that can then transdifferentiate to these ILC1. So we think that the hypothesis that basically HV inflammatory state drives this ILC3s into ILC1 subsets. So to study this in HV infected lymphoid tissue, we have long-term collaboration with the Alfred Hospital in Durban, and particularly with Frank Anderson here, who is head of the GI unit, where he helps us collecting these resected material. And this is really a massive effort, not only from the clinical core in RA, but also from a whole team of nurses at the Alfred Hospital. So we've been collecting these lymph nodes that come from mostly individuals undergoing surgery for pancreas, but also liver resection. And when we perform these surgeries, we also obtain 1 or 2 lymph nodes that sits in these kind of draining compartments, particularly the celiac lymph nodes and the hepatic lymph nodes. We've studied in these data I'm going to show you just now. So this is really like a very defined structure of these lymph nodes that we can then process in different assays here. So first out, we're starting out by looking where's the location of ILCs in lymph nodes, and that's really hasn't been any studies. There's been a little bit done in tonsils because that's kind of easier to access for most people, but the central GI located lymph nodes, we really don't know much about where these ILCs are sitting. So here's CXCR5 staining, and this is really just to define the germinal centers, and we can see this is mostly B cells will be expressing a CXCR5 expression, but clearly shows you where the germinal centers are located here. If you look at the CD117, which is one of the canonical markers for ILC3 expression, we can see it's really located outside the germinal centers here, and basically excluded from these GCs. When we look at the T cells, we can see the T cells sits mostly outside the GCs, except the T follicular helper cells, so the germinal center homing cells here that sits right in the center with this B cell zone around it here. So very defined structures, and we will see that the overlap of RLC3 cells with T cell compartments here. So here's an uninfected donor. We stained for P24, and obviously we saw a negative background here, but again, very clear signal in this donor here about the RLC3 subsets sitting outside the germinal center. When we then look in HCA-infected lymph nodes, we can see P24, which is a protein of the capsid in the H3 particle. We can see that's really located mostly outside the lymphoid follicles. But interestingly enough, this is a participant also fully suppressed in the plasma, but we see lots of P24 levels in tissue. That also sits in the same near proximity to where the RLC3 population is located. So we also performed flow cytometry to identify these cells, and I'm not going to go into the sort of complicated gating here, but really just to say that these cells are very infrequent. They represent less than 1% of lymphocytes in this tissue, but the dominant RLC population is really RLC3 cells that we can identify here, alongside with the NK cells. And here's a little bit of data we've been analyzing a while ago, and we see some signal that the RLC3 population is probably slightly reduced, whereas the NK population here is actually increased. And when we do the NK to RLC3 ratio, we can see HCA-infected individuals really have a skewed ratio, which is normally 1-to-1 in an uninfected lymph node, but in an infected lymph nodes, it's almost like 3-to-1. We have another, I think, 40 or 50 lymph nodes where we are currently analyzing for the same, to see whether this is going to hold up. So to look more into the gene expression and the actual function and what is the impact of HRV on these tissue resident RLC3 subsets in infected lymph nodes, we've been performing this logistical setup where we obtain the tissue, in this case the lymph nodes, from Frank Anderson's clinic, from Abu Hospital, located only 3 to 4 kilometers from our institute. Then they get delivered just early afternoon, processed by Elvira, and then Emmanuel, he's been loading these ones into the sequel assay. And Emmanuel and Jens Akili have been working up the libraries here, and then we've been sequencing those libraries at my affiliation in Copenhagen on a NovaSeq 6000. So the advantage of having this live tissue is really we can then do staining, and we can do live sorting of live cells completely untouched. And then we can enrich for these lineage negative cells, the CD3 negative, CD19 negative, and thereby load these fairly rare cells, less than 1%, into sequel and maintain or generate an atlas of these otherwise cells that you wouldn't see in this assay because they're too infrequent. So when Nicholas, he's performed the sort of the basic UMAP here, this is the kind of cells you see. As you can see, there are no T cells really identified here, only very little compared to what you would see in an untouched data set, but we see quite a few myeloid cells, and we see a number of innate lymphocytes here. So he's basically been subsetting these cells into innate lymphocytes, and finally into sort of more pure RLC3 cells that is dominated by CD117 expression, ROS-C, which is encoding ROR-gamma T, and then IL-1 receptor 1. So these are established sort of RLC3 gene signatures in lymphoid tissue. But this is the first time we see an actual lymph node. So we identify 4 different RLC3 subsets here. And when we look at some of the markers that defines them, we can see IL-7 receptors, which is CD127, expressed in all of them. We don't see any lineage markers expressed as expected. And then we can see some of the core RLC3 markers here. The c-KIT is very dominant. And then, interestingly enough, we see KLRB1 also expressed in all of them. So this really shows us these are true RLC3 cells. And then, interestingly enough, if we look at cluster 3 here, we can see that we have transcription factors encoding Iolis, for example, here coming up, and also in conjunction with some of the NK cell markers. And this is really a strong indicator of these X-RLC3 cells that I showed you in some of the earlier cartoons. And when we just look at the pathways that distinguish these different subsets, we can see that they have very distinct profiles, and particularly TGF-beta pathways comes up in these more like stressed RLC3s and also in the XRLC3s here. So this really shows you that there are multiple different RLC3 subsets in these lymph nodes. And then when we try to sort of look into more of these very few XRLC3 signatures we identify, we can see they're only coming from the HCA-infected lymph nodes on the left-hand side here, whereas we couldn't find these ones in the HCA-uninfected lymph nodes. And when we look at a trajectory analysis, we can see again the transcript here encoding Iolis, which is very key for these XRLC3s. They come up in this cluster here, whereas IL-23 receptor is kind of getting downregulated. So this is the first time we actually identify these XRLC3s in lymph node, and it seems to be, at least in these preliminary data, enriched in the HCA-infected lymph nodes. If we just go back and look at what are the overall transcriptional response of RLC3s in the HCA-infected lymph nodes, we can see on the right-hand side here, this is upregulated in HCA infection, and we can see these ribosomal genes are highly up in our infected tissue, suggesting that there's a activation of gene transcription and translation going on in these subsets here. We have downregulation of some of the HLA-C, CXCR4, HLA, and c-KIT. And when we look into sort of the difference between HCA-infected, the red, and HCA-uninfected, the green ones, we can see the infected lymph nodes with RLC3s has way more genes expressed and also higher transcript level than the uninfected lymph nodes. And if we look at some of the other genes that are important here, we see ribosomal genes highly upregulated 10 to the minus 58, so highly significant here. Genes involved in cytoskeleton also upregulated. Genes involved in interstellar processing also up, and particularly also actually, interesting enough, IL-7 receptor, which is one of the key markers for RLCs in general, is also upregulated in the infected lymph nodes. If you look at some of the other genes that are regulated, we see c-KIT, which is CD117. That was the marker I showed you in the histology just earlier, and we can see that's actually getting downregulated, and we think that could potentially be a sign of this RLC3 to RLC1 differentiation that is currently undergoing in these populations here. Other genes like IL-23 receptors are really important to sense IL-23 production from the myeloid subsets, which we think are the sort of sentinel for detection of potential microbial products in this lymphoid tissue. Other interesting things are really TGF-beta that I'm going to focus a little bit on here, which we can see also. We see a significant upregulation of TGF-beta, and we think that could potentially be involved in this fibroblast activation and collagen deposition. Then we also have CD69, which you may know as a tissue-resident marker, and we can see that it's actually lost in these RLC3 subsets. So we wonder whether this is actually a surrogate marker for RLC3 loss, or maybe even for recirculation of RLC3 cells in HCA infection. If you just look at some of the pathways, we can see comparing HCA negative and HCA positive RLC3 subsets, we can really see a very clear signal here, and particularly TGF-beta is one of our interest pathways coming up here, where we believe that could potentially be involved in this fibrosis. JAK-STAT pathways are also coming up as a significant pathways. And when we look at the IPA pathways and see basically what is the associated pathways looking at these RLC3 genes, we see infectious diseases are one of the top scoring one. Cell death, movement of cells, we see cellular development, growth and proliferation, and particularly also inflammatory response and inflammatory diseases. So all of these pathways really suggest that these RLC3 cells are playing a pretty important role in the disease in these HCA-infected lymph nodes. So here's just another analysis tool from IPA. And again, when we look at where's the sort of network focused, and again, TGF-beta1 is really at the center of this network of the genes that are regulated in these type 3 RLCs in lymph nodes. And we can see they're sort of linked up to trafficking, stimulation of T cells, and proliferation, and also activation of fibroblasts. So we're quite interested in sort of studying a bit further what happens to what is the contribution of these RLC3 cells to lymph node fibrosis in HCA-infected lymph nodes. So to do this, we've also gone back to another data set from the same lymph nodes where we're looking at untouched cells. So we haven't enriched for any cells here. So here we're dominated by T and B cells, and we basically currently trying to look into the TGF-beta production in these cells. And what we can see in the HCV negative individuals, we don't see any difference between TGF-beta production between RLC3 cells and other TGF-beta producing cells. But if you look at the HCV infected lymph nodes, we can see a significant downregulation of TGF-beta in these RLC3 populations in lymph nodes. So this is a very preliminary work in these untouched data sets that we're currently analyzing here. So I just want to finish off with our hypothesis and a proposed mechanisms what could actually lead to or contribute to inflammation in the lymphoid tissue, and in this case, the lymph node draining the hepatic and celiac lymph nodes, where we see this HV-induced inflamed lymph node here, provided the microenvironment for activation. We see a lot of IL-1 beta, a lot of IL-12, IL-32 produced from our marlet population, and they could be involved in actually driving these conventional RLC3 cells into NK-like RLC3 cells, and also finally into this more like XRLC3 NK-like phenotype that are enriched for granzyme A and perforin expression. And then the other pathways here is really the conventional RLC3 subsets could also play a role in particularly TGF-beta production and contribution of fibrosis. So we want to see actually what is the relative contribution of TGF-beta from these cells compared to other immune subsets that are also TGF-beta producing cells. So to perform these data, it's really a big collaboration between members of my own lab, and Nicolas has been driving the analysis. Emmanuel and Rabi has been performing a lot of the wet lab stuff. Clinical core from Farina is really excellent in maintaining these collaborations with the surgical units, and also making sure logistics is working on the ground, combined with Fuji, our research nurse. Other members, and particularly members of the Shalik lab, have been supporting the implementation of single-cell RNA sequencing, and of course the GI unit from particularly Frank Anderson, and then also the sequence core at University of Copenhagen also helped in this project.